joining us for the afternoon session. I'm pleased to introduce Ilan Hadar, Chairman and CEO of Silexion Therapeutics, a clinical stage biotech company developing RNA interference therapies for some of the most aggressive and treatment-resistant cancers. Silexion came public in August of 2024 and now trades on NASDAQ under the ticker SLXN. The company's lead focus is on KRAS-mutated cancers, specifically pancreatic, colorectal, and non-small cell lung cancer, all areas of significant unmet need. Silexion's approach uses siRNA technology to silence mutated KRAS genes before the cancer-causing protein is produced, in contrast to small molecule inhibitors that try to block the protein after it's already active. This upstream targeting has the potential to overcome resistance, improve tolerability, and achieve deeper, longer-lasting responses. The company's first-generation therapy, LODR, showed a 56% objective response rate and a 9.3% survival benefit in a phase 2 study in patients with locally advanced pancreatic cancer. Now the team is advancing SIL-204, a next-generation long-acting siRNA with broad KRAS mutation coverage, which we'll talk about today. Please join me in welcoming Ilan Hadar.
My name is Ilan, as Eric mentioned, and I'm the Chairman and CEO of Silexion Therapeutics Corp. It's a Caymanese-based company with the headquarters, with the executive team located in Tel Aviv, Israel. With me today, I have my CFO here of the company. She joined me here today. I'm going to use forecast. I'm going to say I believe, we hope, so we do need to present the forward-looking statement. And with that, I will jump into the presentation. So we have developed siRNA platform for treating various types of cancer, but we have chosen to go with pancreatic cancer because we think that's really where there is a real need. And we believe that we can give some hope to those patients. The company, before my time, ran a study with a product called LODR. It was, the target was for KRAS 12D and V mutated patients. We will discuss that trial. But when I joined the company back in April 2022 and I looked at the data and the asset, I came to the conclusion that the LODR cannot become a commercial asset, and I will discuss the reason and how we improved it and what we did. And you can see here some of the work we did in the last 2 years. As a small private company, it was clear that we cannot raise the funds required to move forward with the plan. So I've decided to take the company public, and with my CFO here, we did a merger with a SPAC back in August 2024. And from that day, now it's a public information, we raised approximately $15 million, 1.5. And that gives us the runway to continue the development of the drug. And I hope that we will be able to bring it to market as soon as possible. So KRAS, it's known today, and it's validated that KRAS is really responsible for many types of cancer. And you can see the pancreatic cancer here, that 74% is responsible for that cancer with G12D and V, here, colorectal cancer and other types of cancer. So we know today what's causing the cancer, but still you have to treat it, and that's, of course, more challenging. So if we look at the way pancreatic cancer develops, usually. So if you have it, that's what we call the best stage. If you can detect it at this stage, so it's not locally advanced yet, or what we call borderline resectable pancreatic cancer, because here you probably cannot take it out, touching the blood vessel. And at this stage, when it becomes metastatic, usually the lifespan is really, really short, because it goes to the liver and then to the rest of the body. And as you can see, those patients, really, really short lifespan. So that's why we chose to go to pancreatic cancer and maybe provide some hope to those patients. So So the mechanism of action and why we chose to go with siRNA. And today we announced exciting news. We announced today that we are also going to do systemic treatment and not only intratumor. We put a PR this morning. It's out there, and we updated the presentation. It's in our website, so you can download the presentation later on. And I'm going to discuss the new treatment as well, because we really have exciting data. So when the mutated gene happens, there is no way of going back. It stays in body and cannot really fix it anymore. But you don't want that mutated gene to continue to produce the protein that causes the cancer, causes the cells to grow. And what we try to do, we try to insert the siRNA. First of all, it was intratumor, and then we decided to do it also systemic, in hope to stop the cancer from growing and tell the body to act with a functional mutated KRAS protein and really stop growing the cancer. Maybe the cancer will start to shrink, then maybe we can get it out. And our goal is to put as much as possible siRNA into the body, hoping that it will do the work and tell the body to stop growing the cancer. That's the mechanism of action, what we do. And I think we proved the proof of concept in the phase study that we have conducted. There is competition out there, mainly with KRAS inhibitors. We believe that siRNA is the better way to go. We had dinner last night and we discussed the side effects of those treatments, the rash and other issues that those patients have. And we believe that siRNA is much more safe and there will be much less side effects. And we think that we will come with a better solution. So, as I said, treatment resistance, low tolerability, adverse events, and limited overall response. And we hope we will be able to prove that our product will be better when we go into the trial and hopefully to commercialize it. Numbers are huge. I don't need to present the numbers. If you can come with a working drug for KRAS, many types of KRAS, numbers are out there, and I'm sure any company that will come with such a drug will be able to show revenues in hundreds of millions. So the first trial that the company conducted, the phase 2 trial, that trial began around 2018, as I said, before my time. I've concluded the trial back in 2023. So the trial, the proof of concept trial, we had two cohorts really. Cohort 1 was with control arm and treating the LODER with the standard of care. That's the chemotherapy. In total, 29 patients went through cohort 1. And then moving to cohort 2 as chemotherapy changed. They moved from one type to another type, so we also had to adapt, and the trial design changed. And another 20 patients came into cohort 2. Endpoints, of course, were overall survival, response rate, safety, and tolerability. Here you can see some data from the trial. Mainly the trial was conducted in the US, 4 sites, 62% of the treated patients in the US, and also in Israel, 5 sites. The leading sites in Israel is Sheba Medical Center, and I will discuss Sheba shortly. And we tried to treat the locally advanced patients and also the non-resectable in cohort 2. And you can see here the data, mainly DNV, KRAS G12 DNV, but also others came into the trial. And looking at the results, and that's what really impressed me and the team when we joined, as I said, approximately 3 years ago. So most of the patients, this is cohort 1 and 2 combined, most of the patients, the tumor shrank. And where you see those patients went into surgery to take the tumor out. And you can see the results. Of course, it's not statistically significant as it was a small trial. It never meant to be statistically significant, but we saw that those patients did respond to the treatment, and really the tumor shrank. But the sad story is that those patients did not die from the pancreatic tumor treatment. Eventually, that tumor went into the metastatic phase, and they continued to die. So what do you do? And that's exactly what we were thinking, how we can advance the treatment, maybe to treat also the metastatic phase. And that's what really we did in the last year, and I will present exactly what we are doing. So we were impressed. We were impressed, and we thought maybe siRNA is working. Maybe we do have something. Also, you can see here that the life expectancy of those patients increased by, on average, approximately 10 months. It's nice, from 13.4 months, 13 months on average. That's also from literature, the expected life expectancy. And it moved to 23 months, and again, it's a proof of concept study, but it's a good sign. It's a good sign when you see such a curve. With no really side effects, side events, or serious adverse events, and we reported all the numbers, and you can see here that from the siRNA, really there are no there are no adverse events. It's really from the treatment, from putting the loader, the implants, and so forth, and also the investigators that reported that the loader treatment was well tolerated. The DSMB didn't have any concern about the treatment. But something that we did notice, that the loader was really not detected in the body during the trial. When we tried to detect it to see if it's still there, it wasn't. And that's also something that I was concerned about, that we are not sure for how long it is treating and giving an effect. So what we did, we have decided to do two things, basically. First of all, to enhance stability, and I will show the data. And then also to try and broaden the effect of the siRNA on other types of cancers as well. And that gave us the hope that we can do a systemic treatment as well, and not only the intra-tumor administration. And I must say that I think that we have succeeded with that task, and we announced, as I said today, this morning, we announced that we are broadening the trial, the coming trial, to do an intra-tumor treatment and also systemic treatment as well. That will be subcutaneously administered, probably once a month. Probably, maybe we can do a couple of months, every couple of months, but for now, 1 month, and we hope that also to give treatment to the systemic phase of the disease. So here you can really see the advantages of SIL-204 over loader, and we really amplified the activity of our siRNA, and we showed that. And the most important thing, that stability went up from few minutes in human plasma over 48 hours, and now also when we are treating mice at this stage, we can find the siRNA in the body, in the organs, after 56 days. That's really encouraging, because that's really what you want. You want the siRNA to stay in the body as long as possible, to go around, stay there, and maybe fight the cancer. We also added the hydrophobic lead to our siRNA, so it will have better access into the cells. As I mentioned, subcutaneously administration. That's something that we also added, and for the first time I'm presenting it. We want to really rush into clinic as soon as possible, but we still have some work to do, what we are doing this year. So this is really interesting. So we try to increase coverage of KRAS mutations. As I said, the loader was only covering D and V, so we decided to do the wild type as well. D, V, you can see here maybe Q61H, G13D, and that in the future will give us the ability to treat other kinds of cancer, and not only to stay in pancreatic cancer. And we did it using our partners. We are really working with a good company developing the siRNA. They are located here in the US. They will manufacture the siRNA for us. And with that, we really believe that we have something that can work on other types of cancer in the future. And as I mentioned, that's really important for us, stability-wise. So siRNA, the low there, less than an hour, disappeared from the body. And with SIL-204, over 48 hours, and you can see really the same level stay. And on top of that, and that's really important because we don't have to do any more extended release mechanism. We can just inject it in a solution, and it will stay in the body. And what was really encouraging, that we also found it in mice, and you can see here the organs after 56 days. 56 days, those mice, they didn't die from the siRNA, but still siRNA was in the body. And that's really encouraging because with doing that, maybe we can fight the metastatic phase and give some more hope to those patients. Another study we ran was with chemotherapy to check if we have any advantages over the standard of care, the chemotherapy. And not really surprising, but you can see that some chemotherapy has no effect. Really no effect therapy. But when we added SIL-204, we can really see something is happening. Something is happening and tumors do shrink. And you can also see it here. Untreated, only chemo, and with SIL-204. And you can see it's really encouraging. And you can see that everything is statistically significant, highly statistically significant. That's again a very good sign for us, and we hope that we can really repeat the same in human when we start the trial. And we ran many more models. Here we took pancreatic cancer and xenographed them into mice, and again you can see the control, and you can see SIL-204. You can see it's still here. You can see it here, average tumor area day 15, and you can see the necrosis. Again, highly statistically significant. And the numbers do show that probably our siRNA is doing something. It's doing something. And we are running the models again and again, and again we are being surprised by the results, and also the investigators that are seeing the results. And we are working with Hadar, a medical center in Israel, and they see it and they say, wow, that's really encouraging. And we have some more, and I can present some more and more models. And all of that data will be used, of course, when we approach the regulatory authorities later this year and ask to initiate the trial. You can see here again that in the liver really disappeared, tumor. That's really encouraging. Again, intestine, same results. And that's also the systemic administration and why we highly believe that we need to go in that route as well, to do the systemic treatment with no negative control, what we call, and again, SIL, a low dose. And you can see that the tumors are really not growing. And some more encouraging data showing the same. Here's the plan. That's what we plan to do. So we met all the milestones. We promised when we went public. Right now we are working on manufacturing the siRNA, GMP siRNA for treating humans. We are working with another partner working on the solution. The second half of this year we will meet the regulatory authorities in Germany and in Israel. We will submit the CTA at the beginning of 2026 to the EU authorities. But it seems now that we will initiate the trial in Israel before we will initiate it in Europe, because Sheba Medical Center, and really we are working with one of the leading, let me jump here, we are working with one of the leading investigators for pancreatic cancer, Dr. from Israel, and you can see the rest of the names. But she said to us, just bringing me the GMP material, show me that the tox is good, it's safe, and I want to start administrating people as soon as possible. I don't want to wait. So it's maybe a good plan for us to start to go in that route, and first of all to initiate the trial in Israel, then expand to Europe. Also, the German authorities also said, bring us the material, show us that it is safe, and based on the phase 2 you conducted, we want you to initiate the trial as soon as possible. And after we have the data, it's not really the regular path. We will approach the FDA, because the FDA is really strict. The FDA, they have their own way showing phase 1 studies, tox studies, PK studies, but we don't have the time. We don't have the time to do all of those studies. We want to initiate as soon as possible, so we will do Israel, EU, and then with the first 15 patients, we will approach the FDA, show the safety data, and we'll ask to expand it to the US. If all goes well, that study will probably take a few years, but I hope that by 2029, 2030, we will have really a ready asset to become a commercial asset. So to summarize, we did the LODER study. As I said, we stopped developing the LODER, moved to SIL-204 using the safety data from the LODER. And if we will have more equity, we will move to other types of KRAS mutated cancer types. And I highly believe that we can also have some offering there. Of course, IP, our siRNA is protected. We have a couple of patents really covering that. Those patents are going into 2045. Already showed the slide. We are working with the best investigators, and they all really want to bring them the siRNA, and they want to start the trials. That's the team leading the company. As you can all see, we all met at Formix. Dr. Mitchell Shirvan is really the scientist, the mind. He came from Teva, 25 years in Teva developing drugs. Dr. Shirvan really launched many drugs in the US. I only launched a couple while I was at Formix, and also Mirit. You can still see one of the drugs I've launched. Look for Amzik here in the US. You will find it for treating acne. So that's really the team, and we are located in Tel Aviv, Israel. So with that, I will be more than happy to take any questions.